My name is Sabra Klein. I am one of the co-organizers of this webinar. I am thrilled to be here. I think we've heard just some amazing discussions and sharing of data, not all of which are showing us identical patterns. I think we've learned a lot about how BCG in particular and both the dose, so the number of vaccinations, as well as the strain, even whether there is a visible scar or no scar from other vaccines. I think we have a lot to discuss about whether the use of live versus inactivated vaccines, as well as even the sex and age of the participants, or even the age of people when they first are exposed to BCG, could all be playing a role in whether or not we see protective effects of these vaccines in terms of non-specific effects on COVID-19 outcomes. And I would love to do some introductions of our panel speakers who are going to work together to synthesize what we have been discussing thus far, and maybe even pose additional questions or considerations as we all move forward in this important area of non-specific effects of vaccines. So I would like to welcome Professor Nigel Curtis from the University of Melbourne and the Murdoch Children's Research Institute, Dr. Konstantin Chumakov, who is Associate Director of the US FDA Office of Vaccines, Dr. Eva Kaufman, Assistant Professor at Queen's University in Kingston, Ontario in Canada, Dr. Eleanor Fish, a professor in the Department of Immunology at the University of Toronto and scientist at Toronto General Hospital Research Institute in Toronto, Canada. And finally, our fearless leader, Professor Christine Stabell Benn, Professor of Global Health at the Bandom Health Project and of the University of Southern. And I am sorry, this is what happens on Zoom. So I know Eleanor, I would love to be able to mute myself because somebody's at my door, and let you mentioned you have some specific questions you would like to start this discussion with. So I would welcome you starting us off.
That's great, thank you. I'm hoping I'm able to screen share, because what I'd like to do is just bring up, and I'm hoping we can all see this. It just occurred to me that, you know, as there are throughout all these presentations, there are a number of issues that were raised, and I think these could be a starting point. So I'm just going to whip through them now, and then once perhaps all the other panelists have had an opportunity to make some comments, we can see which ones pop up as perhaps the most pertinent ones to discuss. So it occurs to me that we've had issues raised versus the effects, these non-specific effects of these live vaccines that perhaps are influenced by whether it's an mRNA versus a protein vaccine or any other kind of vaccine, and what are the time intervals of the chronology between these live and non-live vaccines and the effects that we see in terms of COVID outcomes. Mihai very nicely presented data on the role of monocytes, and we've alluded to B cells, but what about, and we've heard also from Denise about T cells, but how can we bring this all together and consider which immune cell populations are perhaps most affected or effect in the context of these non-specific effects? What are the effects of these non-specific effects on long COVID, on MIS-C, that's the childhood illness, which actually probably is going to be Kawasaki disease at the end of the day. We've alluded to sex differences. What's the durability and what's the impact of the chronology and the time intervals on the durability of these effects? Given Mihai's data on BCG-mediated induction of chemokines and pro-inflammatory cytokines, why did we get an exacerbated COVID severity? It's sort of, I'm scratching my head. A key issue which we perhaps haven't been able to address in many of these trials is what are the effects of these live vaccines on post-exposure contact transmission in a household? So yes, there have been studies done certainly in Guinea-Bissau where we've looked at individuals being vaccinated, but what's going on in their households? Do you protect from transmission there? And, you know, again, OPV, MMR effects, you know, how are they modified by prior BCG? So these are some of the things that sort of jumped out at me, and I posed them here for further discussion.
Thank you, Eleanor. So anybody who would like to start to address any of the issues that Eleanor so eloquently raised for us? Maybe Christine or Nigel. I don't know if you have some from some of the work that each of you have been doing.
Then I would absolutely point towards Nigel, because Nigel has been one of the key persons in raising some of the factors that can affect or effect modify the non-specific effects of vaccines. So maybe Nigel, you'll take it from there.
Thank you, Christine. Yes, thank you for inviting me to be part of this panel. It's been a very interesting evening. I will point out it's quite late here in Melbourne. So just if I do kind of just drop off, if somebody could just get a dog barking or something like that to wake me up. That's you, Sabra. I think actually, with your permission, I'll just show you some slides. I think, Christine, you thought it might be a good idea to just address some of those effect modifiers. So I might just kick off with that. Christine set me quite a challenge because she said, look, last year I really liked your talk, and it'd be great if you could just repeat that 30-minute talk in less than 3 minutes. So what you're about to get is quite good value, because you're actually going to get a 30-minute talk in 3 minutes, which is, nobody can argue with that, right? So this is really a summary of a very long talk about what you would consider when interpreting BCG and other trials of off-target effects, and really it's applicable to many trials. But I was thinking about this really in light of what Christine said at the beginning, which I think is very important, which was my fear that it's quite possible, for many of the reasons I'm about to tell you, that we don't find an effect of BCG against COVID, as found in some trials already. And there might be some good reasons for that. And my concern that this would set back the whole field, be a really unfortunate, unintended consequence of all our trials. So in considering this, I would use the kind of PICO framework of trials, so PICO standing for the things I've listed there, and very quickly to go to population. So if we start off with population, you already addressed this, Sabra, which age groups? Of course, most of the existing evidence on BCG comes from work in neonates, and the immune system of neonates is, of course, clearly very different, much more malleable than the immune system in older age groups. And you might argue, or it's not arguable, the immune response in the elderly is very different to other adults. We know there are strong sex effects of off-target effects, as we've seen in the oral polio vaccine trials today, and some of the other things I've listed there. I'm going to have to go quickly to fit this in 3 minutes, but we know from some of Mihai's studies that not every individual might be a responder to trained immunity. Maybe only certain individuals are susceptible to training. He also stands for power, and the dreaded sample size calculation that kills us all. And the rule is always that the sample size is larger than you would expect and that you'd like, and certainly that you can afford. And so when we did the power calculations initially for the BRACE study, we guessed there might be a 4% incidence of severe disease in the control group. And if that were to drop by 33% to 2.7% in the BCG group, the required sample size, I'm afraid you've all covered up the bit I need now with your faces. The required sample size would be 8,062, but if the reduction was only 20%, you would need a sample size of 24,000. So power unfortunately, in most studies, we just don't have the funds to kind of do large enough studies. I is for intervention, and we've talked about BCG strain. Denise addressed that a lot, and there's very good evidence that the strains are different. All of the studies I will highlight that have been done during COVID have used the Danish strain, with the exception of BCG Japan and Denise's study. There's good evidence from Frank Shan's study that BCG Russia, for example, didn't have an effect on neonatal all-cause mortality in intensive care patients in India, whereas BCG Denmark has been very effective in other studies in high mortality settings. We've talked obviously about BCG vaccination versus revaccination. I think that's a very important point we can come back to. Co-administered vaccines, we've talked about influenza, of course, COVID vaccines. And there are a myriad of other potential confounders. And I'll just draw your attention to this review that we put out in 2019, which lists the many different things that have been shown to influence the immune response to vaccination, all of which are potential effect modifiers. So C is control, and I think it was Konstantin, no, it was Andreas who pointed out quite correctly that we kid ourselves if we think we're really doing double-blind randomized controlled trials when using BCG, because everybody gets a score. The vast majority of participants get a scar, and that's really important because that really could influence. We've seen that it influences in one of the trials return rates, and certainly might influence whether people get tested. It may be that those people who've had BCG think, oh no, this cough and the cold and a sore throat and breathlessness and anosmia, it must be something else because I've had BCG and they don't get tested, for example. And there are some, no one here has done that, but some previous studies have used a placebo that's in fact active itself and can be confusing. And of course, the outcome measure, very difficult. We might come back to this in discussion. We've certainly had problems with this in the BRACE trial and how you define COVID, I think, and that was also addressed by Denise. And most important is severity. And again, I'd like to come back to that if we have time, because I think that's been a very big feature of the presentation so far. And finally, T is for time. How long does it take for any effect of BCG on COVID to be measurable? And how long does it last? And just draw your attention to these early studies of BCGs reducing all-cause mortality in high mortality settings, such as Guinea-Bissau and Peter Arby's group's studies here. And these curves separate within a few weeks of having, or even days of BCG administration in neonates. Whereas in contrast, as highlighted by Denise, in adults in her studies in patients with type 1 diabetes, it's not only multiple doses of BCG that is needed, but it is a number of years before you see a separation of those curves. And so it looks like if we can afford it, we need to follow our trial populations for a bit longer. And of course, other vaccines, and Anna mentioned this in terms of, we know that sequencing of vaccines, and particularly flu vaccines and COVID vaccines, might have interfered with any effect of BCG in the trials that we've just looked at. So there you have the 30-minute talk in 3 minutes. And to highlight the ones that I think are most important in considering the trials we've just been looking at, I think, I have always been worried about whether the off-target effects of BCG that we are very strong and very clearly seen in neonates would be applicable to adults whose immune systems are very much more kind of fixed, if you like, as opposed to developing immune system in neonates. I think it's a big problem that we've all used this. We've all put our eggs in the one BCG strain basket. And I think looking at, I think many of us have tried to look at this, but probably all underpowered to look at participants who are being revaccinated as opposed to vaccinated. Those who have been vaccinated, in some countries will have been vaccinated at birth, like our participants in Brazil in the BRACE trial, but others will have been vaccinated as teenagers, and that way may well be different. I think this issue is a really, one we like to brush under the carpet, but the fact is that there may well be different testing in those who have and have not had BCG because they know which group they're in. Severity is a key thing which we've definitely come back to because everybody has highlighted that, and this timing issue and the sequencing. And I think that's it. So those, I think, summarize all the effects modifiers. And I'm sorry I had to rush through it, but Christy made me promise.
Nigel, that was fantastic. So why don't we take apart some of what you just put forth for us? I think that was really helpful. So maybe let's start with your comment about age. And if everybody could weigh in about this concept about whether the first exposure to BCG is in neonates versus adults, and maybe that's a place that we can start the discussion and then move through some of Nigel's highlighted areas.
I would propose that I know that Mihai's obviously got great data from the BCG 300 study and many others, and so probably now you can comment and you'll be able to reassure us that you can train adults. But I think it'd be interesting to hear from you about this issue of whether you can train all adults and responders versus non-responders. And I can certainly just quickly add before we start that in our studies in neonates, in the MISS-BEAR study, many of the immunological effects we see induced by BCG are different to what we see in our studies in adults. And I think there are differences, but I think it'd be interesting to be reassured by Mihai that we haven't been wasting our time and that we can train adults.
Absolutely. So if I may respond to Nigel's question. So there are a couple of aspects that I think are important, and age is absolutely very important. What we see we have a cohort of 300 healthy individuals, which are from an age of 20 to 70 or something like that. And we assess the capacity of BCG to induce train immunity. And there are a couple of things that I would like to put on that. The data are not yet published, but we now see a very clear pattern emerging here. Only approximately 50% of the individuals respond well to BCG vaccination in terms of induction of trained immunity responses. We have seen that previously, but we now see it very clearly in this larger group. Importantly, however, is the fact that the people who respond are those who have a poor immune response in the beginning. So what we are seeing is that there are individuals who, before BCG vaccination, we have performed chromatin accessibility in all these individuals before and after BCG vaccination. Also, we have assessed functionally all of them, obviously, before and after BCG vaccination. And the pattern emerges in which, in the beginning, we have a large variability in the immunological response of innate immune cells in these individuals, which associates very nicely, actually stronger than I would have thought, with chromatin accessibility. And we have individuals who already in the beginning, they very much look like other individuals after BCG vaccination. So they have already a chromatin accessibility profile, which enables them to respond perfectly fine to stimulation before BCG. And those are, in general, the individuals who are non-responders. So BCG tends to work in the people who respond poorly in the beginning, who have a closed accessibility in the beginning. That means also that also in clinical trials, probably half of our individuals already respond good, and we cannot improve their response with BCG. That being said, so this is one important aspect. The second important aspect is we do not see a difference depending on age. So, for example, it's not that the young people have chromatin accessibility, which is low, so they respond better, and the elderly the other way, or whatever. We do not see an effect of age on these responses. Of course, now I have to say that our cohort.
Can I interrupt you just for a moment? Will you clarify that you don't see an effect of age on trained immunity?
Correct.
Because I do think Nigel's point about effects of BCG on non-specific effects on of other infections, just to clarify for our participants.
Yes, so we do not see an effect on age, but we have to be very clear that we start at 20 years of age. And the immune system of a child who was just born one day before is completely different from the young person at 20 years of age. And then but we do see that we can train, but we do not have proper comparison between the newborn children and adults and elderly in the same study. We do see also, and we have seen in the paper that we published in Clinical Infectious Diseases and also with Evangelos in Cell, we do see that actually we can train the elderly. However, where I think that the major difference occurs, and this is something that we have to make very clear, for me, it's very clear now that, well, BCG does not work with everything. It does not mean that if BCG would protect against influenza, let's say, will automatically protect against COVID-19, because other pathways, a different pathophysiology may be at play. And maybe we were just unlucky with the type of pathogen and with the type of vaccine that we have tested. At least we see also at cellular response, when we look at the trained immunity response against influenza and COVID-19 in the elderly, we see very clearly that BCG improves much better the cellular response to influenza than to COVID-19. And the same with EVA and with MAS, we did they did the experimental studies in which we see also very clearly BCG can protect much better in experimental studies against influenza than against COVID-19. So I think that
Wonderful. I'm going to thank you, Mihai, so that we can also allow for others to weigh in and continue to raise awareness about some of the issues that you've, elegantly explained to us. So Eleanor, I think much of what Mihai brought up is relevant to some of the questions that you felt were raised in the discussion. I don't know if you'd like to add something.
Well, actually, I just had a quick question for Mihai. So you talk about these trained effects. Did you see any prevalence in one immune cell subset versus another? I think that's useful because we're seeing, for instance, in COVID that, or SARS-CoV-2, the early CD8 T cell response is really important versus in flu, it might be, more than just the T cell response. So could this be contributing to the different kinds of outcomes we might see with different infections? And have you examined that?
We have not looked in detail on that. We have focused mostly on the general immune responses at this moment, but we are currently performing all these stimulation assays and differences with influenza and with COVID-19 at single cell level. So hopefully in not very long time from now, we will able to answer to that question at the different cell population level.
Oh, thank you. So Christine, Nigel really brought up a wonderful point about the strain of BCG. And could you educate our panelists about some of the importance of this in considering these non-specific effects?
We have done several studies and also building on Nigel's experience from the TB-specific effects of BCG. We've also looked at the non-specific effects of BCG in relation to strain, and we do see strong sex differences in the response. I should say, though, that I think the trials that have been done here during the COVID pandemic have all used the two strains that we believe are most strongly inducing innate immunity and protection against other infections, namely the Japanese and the Danish strain. And I haven't seen studies that show clear differences between exactly those two strains. And I wouldn't think that explanation for the trials we are discussing would go a long way in terms of explaining the heterogeneous results. But actually, I think there are some clear effect modifiers amongst those mentioned by Nigel and also alluded to by Mihai, which could reconcile what appears to
Nigel, it looks like Christine might be frozen, and I know that you had a question. So maybe as we wait for her to read.
Yeah, just in relation to that, I thought it'd be interesting to ask Denise, because
hearing that you can't hear me.
Oh yeah,
oh, you're back.
Okay. Well, I'll just run quickly over what I said. I think we have some common factors which can explain and reconcile what looks like heterogeneous results. And one of them was what Mihai mentioned, which was, I mean, you can't improve on a winning model. So basically, if your immune system is already quite well, you don't win a lot from getting BCG. But we have those two trials which show quite stunning effects of BCG, the Greek trial and the US trial, which were done in vulnerable populations who presumably had a lot to gain from that kick from BCG. So I think that is that's one factor which is worth taking into account as a potential effect modifier rather than strain. And then there's the issue about boosting or non-boosting, because as I understand it, Mihai's elderly study, one of them, the big one, was also done in quite vulnerable populations, and they didn't see an effect. But the Netherlands hasn't used BCG in childhood. So I think the two factors that, you know, how vulnerable are people and is it a booster vaccine or no booster vaccine seem to be really important factors, and which would still be compatible with what I think, Nigel, what we are wondering, would BCG have non-specific effects? In my view, there's no doubt when I look at these 5 trials that it does have, but also that there are conditions which are important to take into account. So these were just 2 cents from me. We've talked a lot about BCG, and we should definitely also cover that, but I know Kostya also have some updates on other oral polio vaccine trials. So I would also—
I think before we do that, I'd like Eva to have an opportunity to share her thoughts, and then we can absolutely move off BCG.
Thank you. Yeah, I would just quickly like to come back to the questions from Eleanor on the immune response, and also what Miha already mentioned. And what we also see in both the studies with the human cells as well as with the animal studies is that BCG has definitely an effect on the cellular response, both BCG subcutaneously and BCG intravenously, which is obviously more strong in the IV vaccination. But the question is then the second stimulation, which might be influenza infection or COVID. How well does this activate this initial response, whether that's innate or adaptive? And what we just see with both human cells as well as in the animals is that there is an initial response that is being activated by influenza. So we see these cytokine changes, etc. But the COVID infection does not induce any beneficial or luckily also not a detrimental immune response. And I think that's important in terms of that we need to look, well, we have differences, but what is the functional outcome of them? And functionally, we don't see differences in those models, at least. But I think what is also important that we treat in those models with a very high dose in order to induce a robust infection. So that might override something that we have in a human clinical trial, that some people might get a lower dose infection, where we have a shift by BCG from a symptomatic infection in a non-vaccinated person to asymptomatic in a vaccinated person. But I think, yeah, what in this whole discussion with immune changes, we really need to see what is the functional outcome. I think this is very important to look for that.
While not on the panel, Evangelos has been patiently waiting to ask a question. So I'd like to welcome him to ask a question, and then we really will switch to discussion of vaccines other than BCG.
Well, because we have discussed a lot about the population at risk, and we have shown benefits from our 2 Greek trials in patients who are elderly, patients who have comorbidities, but the major comorbidity for them was cardiovascular risk and metabolic syndrome. So I'm wondering whether, because we know that these are patients who are, let's say, a bit primed due to their cardiovascular disease for high IL-1 beta production, whether this is the reason why BCG is favoring or these patients, they get favorable responses from BCG vaccination. I would like to know your thoughts on that.
Thank you, Evangelos. Anybody, would anybody like to address his query for us?
I guess I have a thought on that. Sorry, my video's not on, but it says the host has enabled my video, so I'm black, but I'm listening, everybody. Oh, start my video. Okay, I'm really here. Okay, sorry. So it answered that question, and Mejia's data is actually pretty interesting on vulnerable populations versus protected one, because now we got the elderly and now we've got these type 1 diabetics responding. We published last year in Cell Eye Science that people who get type 1 diabetes when they're 40, 50 years old, they're called LADA, okay? They don't get diabetes till they're very old, that those people do not have the glycolysis over methylation of the pathways. And those patients also do not have the immune over methylation of the T regulatory cells. And thus they appear to be protected from type 1 diabetes till they're 40 or 50. But the susceptible ones that really respond to BCG clinically, and we detailed this, are those that do have these methylation, over methylation defects. So I think by using these biomarkers on these different pathways, whether it's the innate immune pathway or it's the Treg pathway or the glycolysis pathway, we might be able to subdivide populations. Now in the LADA patients in the US, they've never gotten BCG, but they've obviously gotten some other environmental hit that we don't know yet that puts them into a category where they don't have the defects that BCG treats. So I think in all these populations, we might be able to stratify with these biomarkers to all cell populations of who's going to be most responsive. And maybe the data we're seeing is if you got TB and you've also had multiple BCG vaccines, BCG is not going to help a lot. So who are really the vulnerable populations as adults or vulnerable populations as newborns? So that's my thought for today.
Thank you, Denise. I know, Nigel, would you like to engage Denise? I know you have some additional thoughts.
I've asked you this before, Denise, but I think it'd be worthwhile people hearing your answer, which is there are obviously two different things in your trial, and we don't know whether it's the fact that you're using BCG Japan, and it's only BCG Japan that has such a strong effect, or whether it's because you give multiple doses. And also, you might want to just talk about the fact that BCG Japan is a much higher dose per se. It may be that even a high dose of BCG Denmark may be as effective as BCG Japan. There are very differences in colony forming units, and there are differences in viability, etc.
Yeah.
So do you really think it's the fact that you have to have multiple doses and wait a long time, or is it just BCG Japan is has something particular?
Okay, you always want the multivariate analysis from a mixed population. But we know from trials with BCG and type 1 diabetics, one vaccine doesn't work. Okay, that's been proven over and over again. So that one's an easy one to nail. The other variable that is really important in these adults is the time from the vaccine to the time it clinically protects. So I showed you the data of the paper we're trying to get published, but I'll share something else with this very special group. Simultaneously, open label, we've had 25 subjects going through the same time period, but they got 2 BCG vaccines by the time COVID hit. So same strain, same underlying conditions, same naive status, and those are not protected from BCG. So they only had like 6 months before the exposure to COVID. So 2 to 3 vaccines prior to the onset of COVID did not provide protection. And that's an N of 25, no protection whatsoever. So in these adults that are totally naive, it may be different if you got a BCG and you're priming now with BCG again, but in these naive adults, a year, 6 months is not enough to show the protection. So the timing, we know we need to get out about 2 years in the naive population. And that corresponds to all these biomarkers in T cells, B cells, monocytes, et cetera. So it's a different time course. Than you guys get away with something much nicer in the neonates because it seems to move a lot faster than these adults. And these are adults like from age 20 to 45, 50. It's not the elderly population we also heard about. Okay.
Thank you, Denise.
So we
Eleanor,
need
would you like
extend
to
our
add?
class to look at COVID-24, is what you're saying.
Yeah. So I think we're going to be able, because we're going to keep following these people. They're still double-blinded, placebo-controlled for about another year and a half. So we're going to get more and more data with the different strains of BCG. And also, as you guys totally recognize, most of them are getting vaccinated 3 times with the COVID and Moderna vaccine. So we're going to see the impact of that on the protection as well. Okay.
Eleanor, would you like to add to this discussion?
I just wanted to ask Denise a couple of really quick questions. One is, no BCG, but all these folks have had MMR way back when.
Yeah.
And secondly, none of us have talked about, the issue, the fact that we know that the type 1 interferon response is really critical to these COVID outcomes. Can you go back to your cohorts and look to see whether that's a distinguishing factor?
We absolutely can, because in all these cohorts, every 6 months, they get CD4 cells or monocytes isolated for RNA-seq, and they get CD4 monocytes isolated for methylation experiments. So we can go back and look at what the genes look like they're expressing. As you know, it's really hard to measure by ELISA. We have serum, but that's like really hard. So I'd rather look at the gene expression level and then be happy to share with you the time course of that if it's really increasing at year 2 and 3.
Eva, do you have something to add to this from your data? You're unmuted.
Sorry, I actually have a different question to Christine, so you might want to finish that discussion.
Okay.
Thank you.
All right, well, go ahead with your question, because I am going to start to move us off of
All right.
BCG.
So I know in these human cohorts, we always have this discussion with the BCG scar. And I was just wondering, has the BCG scar ever really been shown to be associated with a working BCG vaccination? So for example, is people that have a BCG scar, are they TST positive, at least for some time after vaccination, for like 15, 20 years? Does this go down with the people aging, or is it maintained? And is there any association between skin test and BCG scar? Thank you.
There is an association, but generally speaking, the TST response declines very quickly after vaccination in children. So only around 40–50% will have a TST reaction by 2 and 6 months post-vaccination, but the scar remains. There are huge variations in how many get a scar. So in a Guinean population, quite homologous, in the rural areas where technique has not been trained that much, it's maybe only 50% who develop a scar, and in the urban areas with a good vaccination technique, it's up to 98%, which tells us a lot about the scar development is not genetically regulated. It's really very much about getting a correctly applied BCG vaccine. So basically the scar is a marker of a correctly applied BCG vaccine with a high number of viable bacteria, because we also see that the Indian vaccine, the Russian strain, does not develop as many scars or lead to development of as many scars as the other strains. So the combination of a good technique, viable bacteria, leads to a scar, which we then again have shown in numerous studies is an extremely good biomarker for a good chance of surviving. It's associated, having a scar is associated with almost a 50% reduction in all-cause mortality in children.
And then these are the children that also are TST positive a couple of months after, usually.
Many of them will have a scar but not be TST positive. So TST responses are also associated with better survival, but less strongly less consistently than the scar.
Okay, thank you.
Konstantin, thank you for being so patient.
Yes, no problem. Yeah, no, I would actually want to propose one other issue for us to consider in addition to the list that Eleanor presented. And it's more have to do with the public health implications and to put this whole issue into perspective. But before I do, actually, I want to mention 2 studies that were not mentioned today. One is published and one is still under review. The published study was conducted in Iran by Dr. Farokh Habibzadeh. And he actually wanted to look at the effects of OPV on COVID-19 incidents. But he failed to get a permission from their Ministry of Health, so he had to go around this and conduct an observational study. So what he did actually was to look at the incidence of COVID among mothers who had babies who were immunized with OPV. As we all know, OPV vaccine recipients, they excrete huge amounts of vaccine virus in their stool. So mothers inevitably get infected with vaccine poliovirus and essentially passively immunized. And what he found is that there was not a single case of COVID among mothers who had babies who were immunized. There were, I think, a few dozen cases in the age-matched cohort of ladies who did not have babies who were recently immunized. So this was it was not a prospective one, obviously, it was an observational study conducted in 2 different locations, 2 cities in Iran. And it was published in JAMA Reports in 2021. So another study that was conducted in 2020 was in Russia. And what they did actually, it was a prospective study conducted in the region, Kirov region. It's a place somewhere in the Urals region in the central Russia. So they have had 2 cohorts. One was given 1 single dose of bivalent OPV and another was left as a placebo control. So they looked for the overall incidence of respiratory diseases and COVID. There was essentially no effect on the incidence of respiratory diseases in general, but there was about 45% decrease of COVID, lab diagnosed COVID disease among those who received bivalent OPV. So this is now will be published pretty soon, because the reviews that we've got were pretty positive, and so I think that it's quite doable to address some issues. So now the issue that I want to raise actually is has to do with the public health context, because I mean, what we're essentially talking here is the repurposing of existing vaccines to prevent COVID-19 and actually address all emerging diseases, because this is actually very, very important issue for pandemic preparedness. So what we found, unfortunately, it was kind of a revelation to me that it's not that easy, because we got a significant pushback from the international public health authorities for use of OPV. Actually, WHO even published their position that they discourage the use of OPV. And the same issue was raised also with BCG, because I think that public health authorities, rightfully perhaps, they are concerned that this off-label use of vaccines could disrupt their, availability for their primary indication. And this is an important issue. Another aspect why I think it's challenging, I mean, with the best science we can generate, it will be challenging to kind of do this repurposing. It's easy on the surface, but if you think about it, industry will not be really thrilled, because I mean, these vaccines are old. They're not really, there's not much to gain from this, from the standpoint of industry. So I think that what is...
Can I just interrupt for a moment? Can you just clarify for our participants? I think you made a very important point hitting on live versus inactivated or killed vaccines and the WHO stand. And I think that is important. And I would welcome you maybe just clarifying a little bit of that for our participants.
Sure, it's not as much live versus inactivated, because this is a dilemma for OPV. And I think in general, so far, the evidence that we have so far indicate that it's perhaps these non-specific protective effects that are more prevalent among live vaccines. That's
Absolutely.
that's
Thank
my
you.
impression.
Thank
But
you.
the issue was that, for instance, specifically with polio vaccines, is that, as you know, OPV is trying to eradicate poliovirus for like 30 plus years now, and of course they use huge quantities of this vaccine, like over 1 billion doses a year. And of course they're concerned that if all of a sudden everybody will start using OPV, there will be not enough for the eradication program. I mean, I'm sympathetic to this, to their issue. So I think that it's perhaps would be more practical to consider development of vaccine that would be specifically developed not for a particular pathogen to prevent, but rather to boost innate immune responses and be a kind of a toolbox, put it in the toolbox of our pandemic preparedness. And actually, it was very interesting to hear this new tuberculosis vaccine that of course it was developed for tuberculosis. But perhaps we should consider development of biological products, vaccines with a broadly protective effects that would be specifically tailored for a strong induction of innate immune responses. And this perhaps would be—
Eleanor, do you want to engage Konstantin in this discussion? Because you've raised the issues of why are we not thinking more about adjuvants and type 1 interferons? And I think what Konstantin is saying is really very consistent with things that you've commented on.
Yeah, no, I'd love actually Gerald to comment on that. This new vaccine, presumably availability once it's firmly approved and demonstrable activity, I'd love you to be using an adjuvant such as interferon. I mean, there's a publication, and I can't remember, I think it was recently, that they showed using interferon as an adjuvant had a really good effect on influenza. But maybe, what Mihai has said and what Christine also supported, and my colleagues and Nigel, you got a good response, you got a good response. We've got to be targeting those who we know are going to be most vulnerable, and an adjuvant vaccine that offers non-specific effects. Perhaps you can even improve its activity by adding something like a type 1 interferon. Gerald, what are your thoughts?
An interesting idea. I mean, we have the data that VPM-1002 actually induces more type 1 interferon responses anyways from the start, from the get-go. It would be quite interesting to also consider a new strain that, I don't know, is genetically modified, for example, to produce even more interferons. That can be considered. However, you would start from the beginning, and development of such a new vaccine strain can take quite a while. We all know that. So
but I've just add the interferon as an adjuvant. Don't.
Yeah. We will definitely consider that. I mean, it sounds quite interesting. Yeah? We'll go back to the drawing board and see what kind of animal models I can think of. Yeah.
But even with BCG and the antigen-specific vaccine, there's beneficial effects in both animal models and in humans, when you look for this Australian study and the Mexican study.
Eleanor, I love that you mentioned adjuvants, because I think that it is also actually dovetails to what I was talking about, because the new vaccine, I mean, that's not a vaccine. Raul Andino in UCSF, he's working on this defective particles of polio defective particles, and he just published a paper that showed that these vaccine, it can be called vaccine actually, that does not have any specific antigens against polio, but it induces broadly protective reaction in animals, of course. So this is a kind of product that I was talking about. that was tailored specifically, not against a specific vaccine, specific pathogen, but rather to generate this broadly protective innate immunity-based response. And what's remarkable, that he also found that these defective particles, they serve as a strong adjuvant to other vaccines. Perhaps, again, by inducing this innate immune response that helps other vaccines to induce a specific adaptive immune response. So this is, all these issues are really interconnected.
Thank you. Christine.
I think this webinar has been fantastic. It's coming to an end, and I want to ask the moderator for permission to wrap up with some
Permission
summarizing.
granted.